Hey guys, it's Justin again in your microbiology laboratory. So we're going to continue on with the unknown project here. We have successfully isolated a gram-positive and a gram-negative microbe. We put them onto working slants, and we're using those slants to do all of our testing to further characterize and identify our microbes. So next in the testing, what we want to do is check for sulfide production, indole, and motility ability. So SIM media, SIM, sulfide, indole, motility, is a semi-solid media, so it has a little bit of agar in there to keep it solid, so you can see it's not going anywhere when I flip it around. And that's the semi-solidness of this media is going to help us determine if our microbe is motile, or is it able to swim or swarm throughout the media. And that kind of tells us what kind of external features that the cells themselves have, as far as flagella and things of that nature, to help it move around in whatever environments it's in. So we want to do a SIM test on both our positive, gram-positive and gram-negative microbe. And this test's a little different because it's not using an inoculating loop, but rather an inoculating straight loop or a stab like this. You can see that there's no loop on the end of this. It's just a straight needle-looking device. That's what we're going to use to pick up our cells and then inoculate the media with. This test has two parts. After the initial inoculation of it, we let it grow, but then we check for the presence of indole by using a prepared reagent that we drip on top of the media, and if there's a color change, we can determine if it's positive or negative for that test. So that'll come when we check the results of these. Also, to check for hydrogen sulfide production in these tests, a black precipitate will form in the media to tell us if it's producing that hydrogen sulfide byproduct or not. And that's also another characteristic on your list to further identify these microbes. So I'm just going to show you how to properly inoculate these, and then we'll look at the results in 24 hours. Okay, so just like we did with our other tests that we've done so far, we have to do our best to remain sterile and use aseptic techniques. So I'm going to go ahead and remove the cap from my working slant on my gram-positive microbe here. I'm going to sterilize the lip of the tube in the Bunsen burner. I'm going to grab my stab tool here. I'm going to sterilize that in the Bunsen burner. Okay. Now I'm going to grab my SIM media here, remove this cap, and sterilize the lip of this tube in the Bunsen burner. Okay, so now I have both of my tubes and the caps to those tubes all in my left hand, and I'm holding the inoculating straight loop in my right. So I've been sure not to touch any surfaces with this loop, so I know it's still sterile. So now I can go into my slant and just touch some of the cells here. Remember, you don't need a lot. Just going to lightly brush it across the surface of some cells that I can see there. And now I'm going to go into this slant right into the middle of the tube, and as gently as I can press down into the center and then pull straight up. Once you're in there, you don't want to move around too much because you don't want to make a big tear in the media, because you want to see a midline to your inoculation so you can see how far away from the midline the cells are motile. So if you go back and forth too much, it'll be hard to tell if your cells are motile or not. Okay, so I'm going to sterilize my straight loop there, and then sterilize the lips of my tubes in the Bunsen burner. Place the caps back on them. Okay, so we're done with our gram positive. Onto our gram negative. Remove the cap from my slant, sterilize in the Bunsen burner. Get my second sim test here. Sterilize the lip of that in the Bunsen burner. I know my inoculation straight loop has been away from any contaminating surfaces, so it's ready to go. So I'm going to grab some cells from here, and then gently go down into the middle of this tube. Like that. Sterilize the loop. And you can kind of see right down the middle there where I went down with the straight loop. So that looks good. I'm going to sterilize the lips of these tubes in the Bunsen burner again, because we're done with the tests. Okay, so now we're going to incubate these tubes 24 hours at 37 Celsius. We're going to look for a black precipitate in them. We're going to look for motility or swimming or swarming of the cells away from where we stabbed it. And then we're going to drop some indole reagent on top to look for a color change to indicate results for that test. So we will see you on the next video.